And so when looking at tissues, the word histology refers to the study of tissues. And in this exam, you're going to be learning about 19 tissues that you find throughout the body. These are pretty much all the families. There are, I think, 2 missing from the 19 here on this packet, but you will learn 19 of them. And so we're going to separate into 2 labs. You'll learn half today and then half the next class, and then you will have your exam on this. And so when we look at histology, if you were to grab any organ, you were to grab any part of any structure, you can see that we can form planes just like we can of the entire body. Here you're seeing some planes being made of just this tube structure. And then you can see that different visualizations can be made. And then you can see that different planes or different slices on different parts of the same structure will yield different views if you slice it in a different section. And so on a microscope, everything is always very thin. It's very, very thin. Like when you look at this segment, you can see that if you were to make a microscope slide of this and you were to take a slice, it would look like this. You'd be missing the rest of it, right? You can't really see the rest of the tube inside, but you're seeing, again, a slice made of this segment here, and that's what you're seeing here. And so it's a two-dimensional visualization that you're typically seeing when you're looking down a microscope. And so keep that in mind, that you are looking at 2D images of something that it technically is 3-dimensional. And so you can see that, for example, if you were to grab a bone and you were to make a section, a longitudinal section, or a sagittal section of it, you would be able to see half of the bone because you would remove one half, and then you can see the inside of the bone itself. You can see that if you were to do a transverse section of the same bone, you would be able to look straight down it as if you were looking at a tree that got chopped. As you know, you'd be looking down at that segment, you'd be looking down into that bone. And then oblique, again, I said that we're not really going to see this one often, and even today we're not really going to see it often. But otherwise, oblique is going to be at an angle, so you can see that it narrows it because it sliced it at an angle as opposed to a perfect x-axis. And so a lot of the structures that you're looking at, and something that makes it easier, is blood vessels. We all have a general idea that blood vessels function like cables. Like, if you were to make a slice of a cable, it's sort of like the vessels in our body. And here you can imagine that this is a blood vessel. If you were to try to make a microscopic image of it, you would take a 2-dimensional slice, and you can see that you would only get the walls of it. You wouldn't get the actual edges of the rest of it because, again, you're only grabbing a slight plane, a slight slice of just this. You're not able to capture the rest of it. And so you can see that that's why it's important to be able to distinguish what you're looking at. And so you can see if you make a transverse, again, you're seeing the inside of that tube pathway there. And so with that, let's get started. The rest of it's not important. And so for your practical, I'll show you some examples of questions that will come up. But otherwise, you'll have to be able to ID the tissue, meaning that a photo will come up and it'll say, what is the name of this tissue? You will have to know a location for this tissue, where it's found in the body. And you will have to know the little specifics about it. So I'm going to write a specifics right here, specifics. Details, because they all have their own unique identities, their own unique properties, and it's important to learn them. Such things as every single one of them, remember that I said cells is a generic word, but you have a brain cell, a heart cell, you have a muscle cell, different type of muscle cells, you have a skin cell. They all have the same overall structures. They all have a plasma membrane or a cell membrane and a nucleus, like we sort of saw a little bit earlier. And we're going to see that some of them have unique structures inside of them. And as we go through it, you'll see these things come up. And so the first thing we got to log away is there are 4 families you care about. There are epithelial tissues, connective tissues, muscle tissues, nervous tissues. Your entire body is made out of these components. They're made out of epithelial tissue, connective tissue, muscle tissue, and nervous tissue. If you were to make a Lego box set of a human being, these would be the 4 boxes that you would need. You would need one of epithelial, connective, muscle, and nervous. So the whole body is made out of some combination of these muscles. Of these tissues, pardon. And so we're going to start with epithelial tissue because epithelial tissue is the harder one today, but the easiest one of the easiest ones to learn. And so epithelial tissue, epi, what does epi mean? Outer or upon. And so this tissue is always found on the surface of things. On the surface, meaning even your skin cells, the first thing that you are looking at are epithelial cells. They're the surface. But even the inner lining, right? When we say the lining of the layers, if you were to look at the inside of the cavities inside of a person, that inner lining, like going down your throat, going down your esophagus, right? Going down that tube that we see on this placard down here. If you were a little piece of food or we were able to magic school bus, I don't know if I'm dating myself here, but if you're I don't know if you ever saw, whenever I say magic school bus, that means that we shrink down and then we're to go inside into the body. And if we were traveling down this esophagus inside of it, it's also made of epithelial tissue. And even inside of the stomach, epithelial tissue. So you can see that you have 2 degrees of epithelial tissue. It's found on the outside of things, and it's found in the inside of things. And so that's what that definition is. And the best thing to hold on to is to think about this esophagus, this worm-like structure that the inside is made out of epithelial tissue and the outside. So it serves as a lining, either on the outside, on the inside. If it's still a little difficult to grasp, just think about your phones, for example, right? If you think about your organs as complex structures, right? The phone is very complex, but the outer encasement that protects my phone, right? That would be an example of epithelial tissue around an organ because it protects it or serves as the surface for it. And so epithelial tissues are essentially that thing. They're layers on the outside, on the surface, and on the inside. And they have a very important role. You'll notice that as I'm walking around or I'm running, I'm not losing water from the inside of my body. It's not like dripping out of me, or when I scratch something, I don't just start leaking blood, leaking water. And that's because these cells, or epithelial cells, are tightly bound together, like floor tiles. Even right now, as you look at the floor tiles right now, as you see this, they sort of resemble this image that I have up here. This is an individual cell that you're seeing highlighted here, with its respective nucleus. Here you're seeing another one. And so you can see that there's no real space in between them at all. So just like the tiles on the floor right now, you can see that there's these little cracks, right, that separate each tile. And so this is the boundary of one cell. And again, they are very tightly bound so that nothing is really escaping them. And so that's a property that we're going to see of epithelial cells that we don't see in a lot of the other cells. And underneath every single, this is going to be a key word here for all of us, basement membrane. Just like the tiles on the ground that we're seeing right now, there is something underneath it keeping them sort of anchored to the floor, to the wood underneath. And that something could be like cement more than likely, or some form of, more than likely it's cement. And so the cement is what's keeping it glued to the ground so that way they're not shifting around and moving around. And so a basement membrane, if you look at another example here, you're seeing our cells. And once more, they are very tightly bound together. You're not seeing a lot of space through here. You're not really going to get through there. And then you can see that at the very base, they have a basement membrane. And no matter how complex, because here you're only seeing one little layer, one single row, and here you're seeing more rows stacked on top of each other, but no matter what, there will always be one layer anchored to the body, and it's going to be, again, anchored by this basement membrane. And so one last thing to add to even to this slide is that following, or even the previous one, is that after the basement membrane, after will follow connective tissue. And get a thicker marker. And remember, connective tissue is another branch or another family, which is not today's topic, but otherwise connective tissue is going to be underneath epithelial tissue. And doesn't that make sense a little bit, right? If this is called connective, it connects things. So the epithelial tissue needs something underneath it to keep it connected and well maintained. And one thing I want you to also add to this page as well is that as you're seeing this purple line, that is your basement membrane. So that means that underneath you have, again, connective tissue, which here is sort of demonstrated by this light pink color here. And then again on this one, you have a basement membrane, and again, our little formula, we have epithelial tissue, basement membrane, and then connective tissue. It always has this general formula. If you can understand that now, it'll save you a lot of complex visualization or understanding a little bit later. But just keep that in mind, you always have epithelial, basement, connective. Epithelial, basement, connective. And so with that, next fact. No blood vessels. It does not directly have blood vessels. We typically refer to anything that doesn't have blood vessels as avascular. Avascular. When we say something is vascular, it means that it has blood supply. And when we say avascular, it means it does not. Sort of like using moral when someone is a moral person, versus when someone is amoral, it means that they don't have morals. So it's the same thing. In Latin, typically, you'll see that play with the letter A, that if it's added to the beginning of a word, it implies the absence of something. And so, that is the role, or at the end of the day, that's why we have connective tissue. Because these cells themselves, remember that they require 4 organic compounds. They need carbohydrates, lipids, proteins, and nucleic acid. We've only talked about 2 so far, carbohydrates and lipids, but they need it, right? Lipids make up the cell membrane. And we saw carbohydrates are a source of energy for these cells. It goes inside and helps them make energy. But if they don't have blood vessels, how does the digestive system deliver it to them then? And the answer lies with connective tissue, which we'll learn a little bit more in detail on Monday. But otherwise, blood vessels, or maybe I'll add it today, and then we'll do the other ones next week. But otherwise, they bring blood vessels, and then they bring blood supply to the segment that's rich in nutrients, and then the nutrients make their way into the cells, and that's why these cells will be able to still survive and grow without the actual direct connections like with blood vessels. And so that's why they're typically referred to as avascular. They do not have blood vessels. They rely on the connective tissue layer to supply them with everything they need to survive. And so one of my favorite things about this particular type of tissue is that it's really easy to identify and learn, generally speaking, because it's named after 2 things, the layers and the shape. And let's just get into it to make our lives a little bit easier. And I just want to give you a quick refresher on this. Some new words probably, actually. Remember that the cell membrane is the outer wall of the cell. That's it for now. It's more complex than that, but we'll address that in lecture. And we have our nucleus, which is the headquarters of each and every individual cell. And then the space outside. All this space that's within the boundaries of that cell membrane, and that is not the nucleus. You can see that I'm not scribbling into the nucleus. All of that is referred to as the cytoplasm. And so in the cytoplasm, this is the best, sort of best thinking about it, like the area around nucleus. That is the clearest definition that you can have for the cytoplasm. The area around the nucleus. And again, it would be all of this. All of that space. And so you can see every single cell has its own cytoplasm. It doesn't really share the cytoplasm, like this cytoplasm isn't shared with this one. They are technically capable of sharing substances between each other, passaging through them, but again, that's not a topic for today. We just keeping it simple. The inside, or the area around the nucleus, is called the cytoplasm. And so, naming-wise, you can see that here, this is a very easy way of understanding. Whenever they're called simple, they have 1 layer. That's it. You can see all the nucleus, or the nuclei, they all arrange themselves in 1 even row. Simple. Stratified means to be layered, more than 1 layer. And this can be much more complicated. Some of them might only have 2 layers. Some of them might have almost roughly 20 in some instances. It really depends on what we're looking at. But whenever we call anything stratified, the implication is that it's a layer stacked upon another layer. But remember, there's only one basement membrane at the lowest layer. There's not a basement membrane here. There isn't one here, and there won't be another one. There is only one basement membrane anchoring the entire stack of stratified tissues. And then, so that's one aspect, right? We said that they're named after their layering, and so simple and stratified. So we have to know those words. And then shape. Squamous, in Latin, well, technically we're just going to keep it to what it actually implies. It's flat. I think it means scale in Latin, so it looks scaly. But otherwise, it's a flat cell. And so you can see that this is a much more smaller cell, right? This is a side view, a lateral view. And this is a view of looking at it from a superior view. So this would be inferior. This would be superior. And lateral, this would be the side. And you can see that they rotate it a little bit so that you can see the top, the superior portion. Here's the lateral portion, right here, the edges here. And then the inferior part would be under there, and you can't really see it. A cuboidal, very easy to learn this one because it's named after the fact that it sort of looks like a cube. And so you can see that in this image. Here's the lateral view of it. Here's the side view, lateral view. And then you can see that if you were to rotate a little bit, here's that same lateral view from the side. Here's the superior side of it. And then underneath is the inferior. And then finally, columnar, like columns, like of a big building. They have big columns that hold those temples that we used to see for those Greek temples. The columnar cells are going to be, again, the lateral view or the side view of the same three-dimensional cell. And so you're seeing, again, the lateral view, the superior view up here, and the inferior view underneath. And so, our first tissue, we're really just going to think about the naming right now. We think about here simple squamous. Simple implies one layer. Squamous implies a flat cell. So if we were to combine it visually, what does it look like? You can see here you have a flat cell, another flat cell, and you can see that they're arranged in one single layer. I'm going to start over here on this side so you can see one single layer. One single layer. This one really does resemble like tiles on the floor. One single layer. You can see just one, no stacking. And what's this purple thing probably underneath it? The basement membrane. Very good. Let's look at another example of the naming system with some pictures, and we'll get into the details after. Now, you can see simple repeats itself. We expect to see a single layer of cells, a single layer of cells, and then you can see cuboidal. Now we're changing only the shape. So cuboidal mean that they're going to be, instead of being flat cells, they're going to be little cubes stacked in a single layer, and once more with our basement membrane keeping it anchored together. And then the next one. You don't have to keep flipping pages. I'm just trying to drive a point here. And so here, simple columnar. Now we expect a single layer of cells, but now we expect the rectangular cells. And so you can see, again, now we're just seeing a stack of rectangular cells, and they're stacked in an order to each other. And keep in mind that this is a three-dimensional image, right? Here's one columnar cell. Here's another one embedded between, another one. But when it was sliced, you're able to see it, the actual lateral or the side view of a cell. So even the cells themselves have the same orientation or use the same anatomical directions that we know. You have a layer that's on top, which is superior, not really the same words for cells. We'll learn a little bit this week and next week. And then you'll see underneath it here, this is going to be more of the inferior layer. And so the inferior layer will typically have the basement membrane, and then the lateral view is just the side that's in contact with the next door neighbor. And they do have little ways of communicating with each other, side to side with each other. And I'll draw for the recording, side to side to with each other. And again, superior at the very top and an inferior. And we don't really use those terms. You can for cells, but we typically have more appropriate cell anatomical direction words, which we'll learn later. But for now, it's just as good. And now this one is one that you haven't seen. We see columnar, right? Columnar. What's the shape of a columnar cell? Rectangular. And this word, are they going to be in a single layer or are they going to be stacked? So they're going to be stacked because we see what keyword? stratified. But what does the word pseudo mean? That's why it might have been tricky. What does it mean? What does pseudoscience mean? Fake science or false science, right? False or fake. And we'll come back to this page again, so don't worry about getting all this just yet. So pseudo-stratified, so it's saying falsely stacked, and you can see all those nuclei don't arrange as nice as we saw it previously, where we saw the columnar, simple columnar cells like this. You can see they were all in a simple arrangement like this, but you can see pseudo-stratified, that they're stacked, but they're not really well stacked, right? You're seeing like one, they're in a single layer, but the nuclei don't really arrange themselves nicely. This is a very unique tissue, a unique one. And then now the good news is, if you understood me so far, you can, don't worry about that one, is that if you look at the last remaining tissues, we see stratified squamous. Stratified squamous. So you can see now we have the flat cells, but now they're going to be stacked on top of each other. You can see we're going to have stratified. Oh, that's it. Okay, yeah. We take off some tissues in labs, which is kind of nice. And that's it, stratified. You can see that that's an instance of stratified when it's stacked. So let's go back and learn these tissues and where they're found and what they do. All right. And so for simple squamous, now a location is going to be inside of the lungs. Inside of the lungs, and you will find at the very edges, remember that, what were the lungs? What happens at the very tips here? Yes, the gas exchanges. And so the lungs are part of what organ system? Respiratory. And so this is where the exchange, as oxygen travels through, eventually it's going to be at the edges. There's a little bit more detail that goes into that, but for simplicity's sake, at these very tips is where you're going to encounter these structures that pretty much have little sacs that have tiny, simple squamous. Simple squamous. Here you're seeing highlighted, here's one squamous cell. Here's another squamous cell. And the reason let's actually find something. I think I have some notes for this that I can use to show. 1 second. Because that's a tough one to actually visualize like that. So let's find an example. Oh, I see how that works. Okay, and perfect, this one. And so when you look at those tips, this is what I mean by those tips. This is that same little tree that we're looking at. And for example, as gas is traveling through, and at the very tips, you would encounter these little globe-like structures. And this is where we find simple squamous. For now, you just need to know that it's found in the lungs. And if you want to know the specific word, it's called the alveoli of the actual lungs. And so what you're looking at is this. There are actually these little bubbles that you can see as air travels through. You can see there's all these blood vessels. So it loads oxygen onto the blood vessels, and then it will circulate through the blood vessels. But right now, all we care about are these little globes or these little bubbles. And if you were to make a slice or a section of it, it would look like this. And these individual little bubbles are made out of little simple squamous cells. I'm going to try my best to draw it. They're made out of simple squamous cells, the nuclei. Here's simple squamous. And so you can see that every single individual one, there's a cell that makes up the wall, and they're tightly bound together, and they're very thin cells. I do believe I actually do have one last image to share. And here you can see it, just like this. This is what it looks like, each individual little cell that you're looking on the inside. It's a complete little bubble, a complete globe, but they sliced it for you. And then, but if you look closely at the edge, right? If you look at this individual cell, you can see that it's been sliced because we made our little section. And what do we see? We see a little flat cell. Here's the lateral view of it, the side view, with its nucleus right there. And then you can see the one that's adjacent to it, which is right there as well. You can see, again, with its nucleus there. So you can see that there's two little flat cells. And again, they resemble tiles in a way. Tiles. They just happen to be flexible little tiles. And so going back to our slides. Oh, dang it. And so that's what we're looking at. We're looking at only a very small slice or a little piece or the side view, the lateral view of those squamous cells. And so it's not the prettiest picture. And so I want you to think about the size of these now. Which one do you think is great for letting small molecules just travel through it? Cuboidal, that's one guess, right? But what would be your logic as to what would make something make it easier? Would it be easier to travel through this one? Or would it be easier to travel through this? Or would it be easier to travel through this? Right? Squamous, and nice and simple. So squamous is thin, so it's great for gas exchange. Gas exchange. And so that's its function. You find it in the lungs because this tissue specializes in exchanging of small materials. And so you can see allows the passage of materials by diffusion. And the word diffusion, this will matter for later in lecture today, so to intro it right now, you already know what diffusion is. Right now, if I were to bring in a hamburger and I were to place it on this table, eventually my students back there will be able to smell it. That's all diffusion is. If you put something here and the little molecules, little hamburger particles, they're going to travel and they're going to go from an area of high concentration where the burger is, and then they're going to make their way, they're going to travel to all the way to your noses, to your back. That's all diffusion is. Diffusion is the movement of a material. And so that's all diffusion is. And so diffusion just is a fancy word to say the movement of material. All right? And we'll get into a little more detail later for that. And so for now, I think the safest location for you to hold on to is that it's found in the lungs. That's all I need you to know. And blood vessels would be also good to hold on to, because doesn't it make sense that if the lungs load oxygen into the blood, then the blood vessels themselves should have this type of tissue to allow them to pass the oxygen to all the tissues throughout the entire body. But we'll learn those details a little bit later down the line. But other than that, that is simple squamous. Remember, you must learn the name of it, and you must know a location. And I would log away the functions because that's also the lecture material. So this is what's really nice about the first exam for lab and lecture, is that they're the exact same material. So this is something that's important to know. All right. And so this is another great example of a blood vessel. See, the blood vessel's the inside. If you remove and you only look at the inner lining of a blood vessel, you would see that it also is made out of these simple squamous cells. You can see how thin these cells are. It's a little hard, a little blurry on the screen, but you can see that it's just one simple cell. Here's another one. Here's the side view of it. And so, excuse me. And so you can see that some images, you can get two images from it. You can make a slice up here, or you can just look at it like this, right? There's two viewpoints if you're looking down this way, or if you're looking at it directly on the surface. And so if you're looking directly on the surface, it just looks like a bunch of tiles, and all of these cells are flat, but you're only seeing one side of it. You can see how tightly bound they are together. And simple squamous, again, is found in the lungs and also the blood vessels. This is another instance now that if you were to look at it, again, if you were to make a slice and you were looking down it this way, but you make a slice like this, then you can expect that it'll look like this. This is the opening that you're seeing, the opening of that blood vessel. And so you can see that it's made out of squamous cells. Oh, let me highlight this one. Squamous. Here's a clear one. Here's another little clear one. Here's another little clear one. And so one new vocab word to add, we already have cell membrane, we have nucleus, we have the area around the nucleus, the cytoplasm, and now the word lumen. This is a fancy word to say the tunnel inside. If we were magic school bus our way inside here, and we were traveling through here inside of this tunnel, that's a lumen. Even simpler, if you're going down the LAX, you've all seen that large airport tunnel. If you travel through it, you're traveling through the lumen of that tunnel, the lumen. Lumen just means the passageway. Or another example, if you're looking at a paper towel, the inside of that paper towel that you look at, that's a lumen. A lumen is just a passageway on the inside. That's all. And so these are these useful flashcards. This is for those of you that like to draw. And I would recommend drawing just a simple arrangement just to help you understand, or you can just copy these if you'd like. But otherwise, or you don't have to draw at all. But the key, this is more applied for everybody. In case you don't like to draw, there's already one for you so that you can just prepare what you need to know. First, let's talk about the cell type. What is the type of cell? What's the shape of this? Squamous or squamous. So make sure to write your squamous here. Structure at arrow number 2. What do we call this wall? The cell membrane. Nice and easy, right? And then the structure at arrow number 3, what's that in the center of every cell? The nucleus. And you may hear me say the word nuclei. One is plural, and this is the plural. I'm just going to write parentheses P, plural, and this one is singular. So one is just nucleus, many are nuclei. Yeah, and so with that. And then the tissue, this is a little bit of a redundant, but this is supposed to help you with spelling, that's all. And so you really just kind of want to practice your spelling without looking at it. And again, the tissue that it represents is simple squamous. But that brings me to my next point. If it asks you for the tissue, it says, I'm going to rephrase this question. If it says ID, the tissue, the answer is simple squamous. Mm-hmm. Oh, right. There's a new version of this. Okay, what's number 4 say on yours? So the space, what would be the space around the nucleus or the blank space? Cytoplasm. So I would imagine that it points into a space like this. Oh, sorry. Like this, maybe, like this, somewhere along one of these areas? Oh, inside. Okay, so what's that one then? That's the lumen. Yes, the lumen. 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 Here's another one. Here's another one. Another one. And you get the idea. Yes, lumen, lumen, lumen. But if it had asked you, it's not here, but what happens if it asked you about this little, it puts an arrow right here, what would be the answer? Cytoplasm. Cytoplasm. And that is very possible, or a possible exam question. So if it, oh, dang it, pointing to the membrane there. Here we go. Yes, and then that would be an example of where we would ask cytoplasm. And so now the number 5 I would imagine is tissue the drawing represents. And so ID tissue is going to be simple squamous. Simple squamous. But if it asked you ID tissue type, what would be your answer? Epithelial tissue. Epithelial tissue. That's the family it belongs to.
Would the questions be like that?
That is very possible, yes. Epithelial tissue. So those are 2 different types of questions. If it says ID the tissue, it's just asking, what is this? If it says ID the tissue type, this word in your head, you should think family. And how many families of tissues do we have? 4. Good. 4. Well, one of them was epithelial. The next one is, what was the one underneath epithelial tissue? Connective. And then the one that helps move skeleton around? Muscle. And then finally we have nervous. Nervous. So those are the 4 answers to type. If they ask you type, you're either answering epithelial, connective, nervous, or muscle. If they ask you ID the tissue, they just want to know, tell me what this is. And then your answer would be simple squamous. All right. And then a location, what's going to be our easy location for this one? Nice. Lungs, air sacs. I'll accept anything that resembles, indicating the lungs, and also blood vessels I'll take too. Yes. All right, and this is just the first example that I'll do for all of you. Otherwise, I'll let you all work on these independently. We'll go over the answers when you come back. And so I just want to get through the materials for today and get as much on the recording so you can get as much of it on the weekend and get a lot done on the weekend. Okay, so with that in mind, Now we have simple cuboidal, which are simple cuboidal cells. And you can see here, cuboidal cells. And the good news is that their functions are very simple: secretion and absorption. Secretion and absorption. Easy function. And we find them in one place that's going to be more relevant for us coming up when we talk about skin is in the glands. And so glands would be a safe or useful thing to remember because it'll be redundant. You're going to learn it again in integumentary and you'll already know. You'll already know what they're made out of. And so glands, and doesn't that make sense? We all have an idea that glands secrete substances, and so it helps with remembering some certain terms like secretion. Absorption a little tougher, but remember that our kidneys are always going to be able to absorb things back into the system or take things out. And so the kidneys are going to be primarily made out of these structures that contain cuboidal cells like this. And what you're looking at, again, you have to imagine like if it was a straw, like it was cut, and all you're seeing is just one little piece of that straw, that tube that's been cut, and you're seeing the inside of it. This is the inside of that tube, the lumen, once more. It doesn't change. And then nothing else changes. Here's our, what's this structure called? Nucleus. What's the space around called? What is the wall around it called? Cell membrane. And what is the thing, the layer that anchors it? Basement membrane. Rehearse that over and over again. And what's underneath basement membranes? Connective tissue. And you can even see basement membrane. Oh, let me get higher. Basement membrane. And then you can see connective tissue at the very bottom of the tissue. So of the actual cuboidal cells. All of this connected. We haven't even learned what this is yet, so you don't really care. You just know generally it's just connective tissue. What is the tissue type of all of this here? Epithelial. What is the tissue type over here in blue? Connective, underneath the basement membrane. Okay. This is another image with those particular types of answers. This is the image that they even use on the exam. Every exam question is going to be coming from this packet. And that's why you want to take some time to look at it with color at home or on your phones or devices as you're studying through it, through your packets, because we're going to use these exact same pictures and it's going to ask these questions almost in the exact same places. All right?
Wait, so all that packet is going to be in the test?
Yes. Your first lab exam is made out of the packet that you were given on the first day and this packet, and that marks the end of the first lab exam material. And so, let's keep it going. And I'll let you all leave these exercises for you at the end of lab or also for at home to finish and study. And you can make them up into flashcards and just keep practicing and rehearsing. And we'll go over the answers when you come back. Now, simple columnar, the good news about epithelial, and like I said, I really like teaching it because it's easier compared to the other tissues. And one of the things that it does is that it does the same thing as cuboidal cells do. And what do cuboidal cells do? What are the two things that it does? Secretes and absorbs. And so, columnar cells, exact same thing. Absorbs and secretes. You've already learned what all the columnar cells do. Absorbs and secretes. Nice and easy. And a location. In the digestive tract, we absorb nutrients, right? We absorb nutrients. So if you look here, where do we encounter these? We encounter them in our small intestines, as you're seeing here. Even here in the large intestines, as you can note this segment here, you also absorb things. And let's talk about the digestive tract just a little bit for fun. And remember that we said that the stomach breaks things down. It breaks things down. And then it'll send everything broken down into the small intestines, and the small intestines will absorb it and place all those yummy nutrients into the blood vessels. You may also see that there's this picture frame, right? This one's called your large intestine. And what it does is that it stores feces, is that once food has gone through this gauntlet of absorption down in the small intestines, it will eventually make its way into the large intestine. And in the large intestine, you store feces. And feces will still have something that you need. It'll have water and salts. And so your body can also reabsorb or absorb salts and liquid or water back into circulation as well. And so the columnar cells specialize in the digestive tract, that they specialize in the small intestine because they absorb. And then they also absorb in the large intestine as well. They just absorb different things. But that details for a little bit later. I just kind of want you to have a general idea of where these cells play a role. And we also said that columnar cells secrete substances. What does the stomach probably secrete at the end of the day when we think about what's inside of the stomach? Mucus is one thing. What's another thing? And acid. And so columnar cells are in there and they're secreting mucus, they're secreting acid, and even in the small intestine, they're absorbing nutrients and they're also secreting mucus as well. So that's really all they do. They're not really like very technologically advanced little living creatures. They're just absorbing and secreting things. And so, with that, now this image is incredibly difficult to understand because it's just a bad picture. But otherwise, this space inside, what's the space inside of things? Lumen. This would be the lumen. We'll have a better picture coming up in a second. But lumen would be the space on the inside. And then our basement membrane is going to be on the opposite side. Opposite side, the basement membrane. No matter what tissue you were looking at, I'm going to just quickly go back here. You'll notice that the lumen is always going to be on the superior side of the cell. And on the inferior side of a cell, it's going to be attached to the basement membrane. Let's look at this orientation. It might be a little bit easier. Superior, inferior, and so the superior side will be the segment that's in contact with the empty space, right? The lumen. The lumen will never be attached to the basement membrane. It just doesn't make sense. And so the basement membrane is always going to be on the inferior side. And so you can see again the lumen, and then the basement membrane is going to be on the opposite side. I know because I even have our connective tissue layer at that side as well. And so you're seeing one columnar cell, and you're seeing the superior side, and then you're also seeing the inferior side, and that's just one side of it. If you look at the opposite side, you can see we have another columnar cell, and inside is our lumen, and this is the superior side. This is the inferior side of the cell, and another basement membrane underneath, and another layer of connective tissue. This picture may look so confusing, but it's simplified on this one. Don't flip pages just yet. But if you look here, this is what I mean. Columnar cell, lumen. And basement membrane, that's one side. And connective tissue follows right down here, which we don't care about just yet. And then you can see on this other side, again, lumen side, basement membrane side, and connective tissue, CT, connective tissue. And all of this is the lumen. So now in the intestinal tract, what you have to imagine is that if you were to grab this small intestine that you're seeing here, in this segment here, this is like a worm, right? And now if we slice it and you were to look inside, or if you want to think, use your imagination a little bit more, if we were a little piece of breakfast, something that we ate, a little breakfast sandwich, we're traveling inside of the tube, right? Your small intestine is like a worm. It's a tube, but you make a slice of it, you remove that slice, and this is what you would see. And so if we're a little piece of nutrition, we'd be traveling through here. And as we're going through here, these cells, they absorb and secrete. They're going to absorb you into the blood vessels. Absorb you into the blood vessels. And let me do it for the recording. As you're traveling through here, if you're a little piece of yummy nutrients or a breakfast, something you just ate, the nutrients will be absorbed through the columnar cells. That is their job. It'll absorb it and place it into the connective tissue layer where there are blood vessels to transport it to the rest of the body. And one last thing to add, because that's absorbing. What's the other function aside from absorbing? Secretion. Now, I want you to think about if you're a little piece of breakfast sandwich what broke you down? What was the first organ that broke you down with acid? The stomach, right? And that acid inside of the stomach is a pH of 2, which is enough to digest you. If you were to grab that acid and you were to place it on your skin, it would burn right through you. And so these cells, or there's going to be cells associated that secrete mucus the other way. So when we say secrete, we're going in this way. When we say absorb, we're going into the blood vessel layers up top in the connective tissue. And so that's an important idea because secretion of mucus is important, because if you didn't have a layer of mucus on the surface, your stomach acid would digest you. So they secrete mucus in order to protect themselves from your own stomach acids. Right? And so with that, back to where we were. And the good news is that's as complicated as it gets today. So if you've been with me so far, we're almost done. And so for now, when we see simple columnar now, there's going to be types of columnar cells. Remember that we talked about that feline can mean bobcat, could mean lion, could mean tiger, could mean house cat. Columnar cells have different types. They have non-ciliated, and then they have ciliated. So it's really non-ciliated versus ciliated. And do we have a... Oops, sorry. Glitched out a little bit here. Just want to see... I guess we only ask you about ciliated. So, okay. Oh, no, we don't. And so when we say non-ciliated, this means that they do not have cilia. And I know we don't know what cilia is yet, so let's just write so that we understand. Do not have cilia. And I'll explain the difference in a bit. Do not have cilia. Now, if you use a microscope and you look very closely, what you'll always see are these finger-like projections on the lumen side. On the side, that's going to be where all those nutrients are passing through. There are these structures that are called microvilli, which are, again, finger-like structures. And they absorb. That is their function. Finger-like structures that absorb substances. And it makes sense, right, that we're only going to see microvilli on the side that we see the lumen. And then on the bottom side, there are no microvilli. Food is not traveling or in contact with this part of the cell. It's only in contact with this part of the cell. Finger-like structures. And so we see that simple columnars, we already know that they absorb and secrete. How do they absorb? They use microvilli. They absorb using microvilli. And I know your pictures might be very small, and so, but luckily I can make it larger on my tablet here. And so you can see now all together that we see our microvilli on the surface of the cells on this little cartoon animation. They animated on top of an actual picture, but you can see this is what it would look like on a microscope. But if you use a very powerful microscope, they would look like little fingers. And so that's what they're trying to show you here, the little fingers. And so this helps increase surface areas for maximum absorption into the cells, and the cells will then give it to the blood vessels a little bit later down the line. One thing that we encounter are assistant cells, accessory cells, cells that are there to help out the columnar cells, and in this case, we have our goblet cells. These goblet cells specialize only in mucus secretion. We need a lot of mucus, and that's their job, is to help with mucus production. And I misspelled mucus here. Mucus. Wait, that looks worse. I'll fix it right now. And then, but otherwise, I think it comes up right now. How do you spell mucus? O-U-S?
No, U.
U-S, the way I had it the first time?
Yeah.
Okay. It still doesn't look right. It'll come up right now, just to double check. So I'm having one of those days. All right. And so our practice, don't forget, non-ciliated means does not have cilia. We'll talk about cilia in a bit.
Professor,
Ask away.
you know how you're talking about like basal is inferior and hirsolumen is superior? Are
Yes.
you using apical?
Yes, that's what we will learn at the end of this. So just to keep it simple right now, just the top and the bottom, and then we apply those very two words, apical and basal. And we'll actually add it right now after I finish goblet cells. And so when we look at our columnar cells now, remember that we have different types. Oh, pardon. Remember that we have these little assistive cells called goblet cells. Their job is to produce mucus. And yes, M-U-C-U-S. Just doesn't look right to me. But anyways, mucus producing cells. And these are going to be cells that embed themselves inside of the columnar cells. They just, like I said, accessory cells. And they help with the secretion of mucus, so to protect the inner lining of acidity of the stomach acid. And not just that, it helps with for materials that are or nutrients that are traveling through the lumen to dissolve into that mucus, and then it helps with further absorption. And so you find goblet cells in many different places. Right now, where it's applicable, we found it associated with simple columnar cells. They are embedded with those columnar cells. We saw that there's another tissue that's going to come up in a little bit, pseudo-stratified columnar, and they have goblet cells as well. They have little cells embedded. They always look like little tiny, like translucent, meaning clear, cells that embed themselves in between them, and their job is to secrete mucus onto that surface, onto the lumen surface. And so you'll see in a little bit that the other tissues use goblet cells as well. Goblet cells. And this is a nice little schematic of goblet cells. Again, they embed themselves and secrete mucus into the lumen, no matter where you find them. You can see that right now we've learned about the one that has to do with the digestive tract. And so, right now, in our respiratory tract, we've only learned about the alveoli, or the air sacs at the end, that have simple squamous. But if we just look at just the windpipe, the trachea, right, the trachea, this, if you look at the inside of it, you will also encounter goblet cells. And you have mucus that helps trap stuff inside of your airway, and then it'll help trap dangerous pathogens and particles that you encounter in the outside world. And so mucus cells become a part of our study. And this image is just summarizing everything I just said. And the last thing I do need to add before we move on is now microvilli versus cilia. And okay. Pseudostratified columnar epithelia are particularly known for having cilia. Cilia. And cilia are also going to be projections that are on the lumen side. Now, what is the difference? I'm going to write it large for everybody. Cilia versus microvilli. What do microvilli do? Absorb. Cilia do not absorb. Their job is to move substances, to move particularly mucus. Mucus. And anything trapped within that mucus. Cilia. And I'm going to give you a story to help you remember this fact. When you look at the trachea, you may have all encountered somebody who does excessive smoking, and you ever heard them cough if they were chronic smokers, right? It's like a coarse, like very kind of rough cough, coarse cough. And that cough is caused because they paralyze cilia. Cilia. And then the cilia's role is to move mucus. On your day-to-day life, as I'm talking to you right now, there's these little particles of dust, particles of your skin cells probably, of anything that's just traveling in the air, and it goes into my airway, and then they're getting trapped inside of my airway. They're going inside, but the role of my cilia is to trap it on the mucus, and then the cilia, they move it and beat it all the way up. It's even called the mucociliary apparatus, escalator. escalator. It's like an escalator of mucus that moves through the cilia. And so it moves mucus up out my airway, and then I swallow all those particles that I put into my airway, whether they're bacteria, whether they're, again, like I said, particles in the atmosphere, that they come in. Your lungs don't want any of that stuff. Your lungs only want oxygen and so, or gases. And so all those particles get trapped inside of the lumen on the edges of where the cells are, and then the cilia move it all the way up. And then again, it'll go into your stomach instead, into your esophagus, or some of us, what do? We spit it out, hack a loogie, and so you get rid of all that material, and that's the difference between cilia and microvilli. Cilia moves trapped substances in mucus inside of the airway. All right, that's what cilia do. Cilia. And I'm going to give you also this other example. You also encounter this inside of the fallopian tubes. They also are responsible for moving the egg further into the uterus. But I guess some of us may not be familiar with those structures, so maybe it's not the greatest example right now. But this is an example of that. Anything trapped within this lumen side, if it has cilia, the role of the cilia is to cause some sort of movement. And then in the airway, the goblet cells will secrete mucus, the mucus will trap particles, and then the cilia will move it out of the airway. And that's what cilia do, move things.
Professor,
Ask away.
is microvilli exclusively to simple columnar epithelium?
No, they are also found on cuboidal cells as well. And that actually is a great question. If we look at cuboidal cells, what was the function of cuboidal cells? Secretion and absorption. When you hear that word absorption, your brain should jump to what structures? No, structures. I hear the microvilli. Microvilli. Absorption are those finger-like structures that help absorb things.
Can cilia be found on simple columnar?
They can be found on simple columnar as well. Yes, they can. All right, so with that. And then there's even goblet cells. You must be able to recognize goblet cells. Again, role to secrete mucus, and that's really it. And make sure to be able to answer these. My packet's a little different from yours, and I just realized it is because my numbers aren't even right, and I'm pretty sure yours are. So your packet is probably correct, and I will upload the new PDF as soon as I get it from Dr. Meyer. And so with that, now pseudostratified ciliated, now we can read this entire thing, right? Pseudostratified, ciliated instead of versus non-ciliated, but ciliated. And where do we find pseudostratified columnar epithelia cells? In the trachea, right? The windpipe. And again, the role is to move the cilia, is to beat the mucus up so that way you can get rid of trapped particles or particles going into your lungs that you don't need or want. And so before we move on now, back to Isabella's point, now directionality of all these cells. There are two words that matter for us, and it's going to be apical. I'm going to use this picture. Apical and basal. Apical refers, as I was saying, superior, and I was saying inferior. Now, the appropriate terms to think about, the side that's in contact with the lumen, the word is apical. And then the side that's in contact with the basement membrane, let this B-A-S help you remember, basal. B-A, like this, basal. Basal. And then up here, apical. Apical. And so we don't use superior or inferior. You technically could, but it's simpler just to say apical and basal. Apical and basal. And so just quickly evaluating each tissue. I'll give you all a second to make sure you got it. Apical and basal. And it doesn't matter if there's different layers. This side is apical. This side is going to be basal. Nothing changes. Apical, basal. And so if we evaluate each tissue, if we look at the inside, this is where the lumen space was. So what's going to be this surface?
Basement. Cell membrane.
Apical. Apical, because it's the side of the lumen. Basal is really hard on this one. I won't ask you a question like that on this. But otherwise, this one's, it's really hard with simple squamous. Too difficult, I think, personally. But this one I can. If we say here the lumen, so what side is this part of the cell? The apical side of each one. The apical side of this cell, the apical side of this cell, the apical side of this cell. And so what's the side of this side called? Basal. What about this side? Basal. This side? Nice. And so the basal side will always be attached to the basement membrane. And no matter what cells we look at, it is always going to be the same thing. What side is going to be the side with microvilli? The lumen, but it's going to be the lumen. So what's the side called, or the orientation? Apical. And then the side with the basement membrane? Basal, right. And so this one, this is the kind of what summarizes it all. What's this side of the cell? Apical. What's the side up here? And then on this side, it's a different orientation, but what's this side? Apical and then basal, right? Very simple. So the connective tissue is always on what side? On the basal side. The lumen is always on the apical side. Cilia is always on the apical side. And basement membranes are always on the... There's only two options, apical or basal. The basement membrane is always on the basal side. Very good. All right. And so pseudostratified, we went over it with the lungs. Otherwise, there's nothing more to add. And then this is an oddball. Transitional epithelia, named after the fact that it can transition in shape. You find it in, what do these two kidney being structures look like here? The kidneys, and then so they're going to passage urine down into, what is this called? It's in the pelvic cavity. The urinary bladder, it's found in the pelvic cavity. And so this is like a balloon, has the ability to expand or collapse on itself. And so the inner lining of it, the epithelial lining of it, the inner lining, the apical side of all this tissue is going to have a type of tissue that can expand. So it's technically known for just having multi-shaped, being polyhedral. Here, this is going to be the lumen side. This is where urine's going to be at. And then this is going to be where it's anchored to the rest of the organ and the basement membrane and the connective tissue. And again, this will expand. And so we have a picture of that on this next slide, right here. That you can see that this is the same picture as this, or same area as this. And you can see this is collapsed, meaning that there's nothing in there. It's just like a balloon that's just really small. And then when your bladder is getting full, you're like stretching it like rubber bands, right? You can see all these cells sort of change their shape and stretch themselves out. And so collapse means to be sort of like loose, like a loose balloon. Distended means to be stretched. And so you're seeing that this tissue specializes in stretching, changing shapes, transitioning in shape. We refer to some of these cells as with a particular name. If they are at the very top, they're called dome cells. Dome cells. Dome cells. And so you're seeing a dome cell relax, a dome cell stretch. That's what these numbers indicate, 4 and 3. But otherwise, they're just dome cells. One is relaxed, one is stretched. Otherwise, there's nothing more to add here. And this is only found in the urinary system. Yes. Yes. Okay. And to move on and finish up pretty much our tissues for epithelial, we have stratified squamous epithelial. So you can see this is a little bit different, but a lot of themes are the same. One more time for the sake of just further concluding this. What is the membrane at this side called? And it is on the what side of cells? Basal. And so this surface side is going to be called apical. Apical. Remember, it's always apical versus basal, the bottom side. And so, the tissue that we're going to learn about right now, forms the lining of things. This, you're going to see two forms. You're going to see non-keratinized, and then you're going to see keratinized without this non at some point. It's going to come up on the next pages. But let's keep it simple right now. What you're seeing is nothing different from what you've seen before. Squamous cell, and then they are stacked upon each other. So you can see all of these flat cells. You can see that there's flat cells stacked on top of each other, going all the way to the top, all the way to the top. And this tissue specializes in high friction environments. So you're going to encounter, and now let's keep it without getting all technical, is that this is like a layer of armor for the inner lining. So you think about the inside of your esophagus, like your mouth, or just the esophagus for now, because we keep using the example. This esophagus, what goes through here every day?
Food, water.
Food. How often a day? Right? Well, for a lot of it changes, right? Like I eat about 4 times a day. So like if you think about that, you think about this segment, you are always passing food through here. So what it is that it's a layer to absorb that friction. You sort of lose these cells. They sort of go away with your food, and then your stomach digests it and recycles the materials. But these cells will always just keep dividing. So no matter what you do, as you keep encountering friction, these cells just keep replacing the inner lining over and over again. And so you encounter non-keratinized. I'll explain what this word means, but you encounter stratified squamous inside of your inner lining of your esophagus, of the throat, because you're always encountering friction every single day. Another place you encounter it, or you encounter friction, is the rectum, or right here. We all have an idea of what the rectum is going to leave. What comes out of the rectum at the end of the digestive tract? Feces. So you defecate every single day, right? We poop every day, and so you need to keep replacing the inner lining of your rectum. And so that's because, again, feces travels through here, takes these cells with it, and then we're just replacing it over and over again. And then, yes, we'll get into that very specifically in a second. And so these are all squamous cells, and they just get flatter and flatter as they progress up. I know that some of them might look a little cubed, and you might be tempted to answer cubed. Don't confuse yourselves. They just happen to get flatter and flatter as they go. Squamous cells all the way up. They're all considered the cell type squamous. And they don't have any multiple basement membranes. You only ever have one basement membrane and once more connective tissue at the very bottom. Always connective tissue. And so this is all the same things that I talked about. Let's keep it going to finish up the last one. Now, we only mentioned right now non-keratinized, as seen above right here. Stratified squamous epithelial cells, non-keratinized, as opposed to keratinized, right? One is keratinized, one is not keratinized. To make sense of this craziness, when you're looking at it from the side, and so we're going to look at the cartoon image of it first. And if we stay on our non-keratinized, another place that it's encountered, and we are seeing it on this little illustration here at the very bottom, is in the vagina. For the vagina will encounter friction during sexual reproduction or intercourse. And so this is a place where we also encounter this type of tissue. And to make sense of our image here, the apical side is going to be the inside of the vagina here. And again, the basal side is going to have this basement membrane, and it anchors it to structures in the body. And when we look at keratinized, nothing changes. You still have a side that has a basal side, but this one's a little unique because it's just this boundary that you're seeing, this highlighted purple region here, basal. So you might want to draw this in. Let me draw it in for the recording for all of you. Here. Let's pick up green color because I feel make this a little bigger. So all of this would be your basal side. And what's the membrane that's going to be attached to all that green there? The basement membrane. Nothing's changed. And then the opposite side is going to be, what's the opposite of basal side? The apical side. Apical, apical. And so what is now the difference between the two is simply where you find it. In the inner linings, you find non-keratinized. You'll notice that I talked about the esophagus, I talked about the rectum, I talked about the vagina. Those are all structures that are within body cavities. But if we think about the surface, keratinized is found on the skin. So isn't that insane that what's inside of your throat, inside of your esophagus, inside of your rectum, you have the same tissue on your skin. But the difference is that the one that is inside your body is non-keratinized, and the one on the outside is keratinized. And the only thing you need to add to it is what is keratinized. And the simple answer is that one, when it says keratinized, it's waterproof in a way. Waterproof for now. We're going to leave it very simple, waterproof. And so you can see that we don't really need to be waterproof in my esophagus. We don't need to be waterproof in the rectum or the vagina, but we do need to be waterproof because, right, this is my outer layer. I can't be losing liquid in water. And so every single cell that you're seeing on the surface, you're looking at a bunch of squamous cells. You're actually looking at this. This is what it looks like on the surface of your skin. This is superficial, and this is deep. Like if right now they're showing you the foot here, right? The surface, the bottom, the plantar region of your foot, that is going, you're thinking about a nail, like if you step on a nail would go into the surface of your skin, go deep, 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 and then finally that penetrates even further. Ouch, right? It hurts, right? You puncture some nerves and it hurts, you feel it. And so this is the superficial, and as you travel deeper into the tissue, you go deeper into the skin, deeper into the skin. And so that's the difference between keratinized and non-keratinized, is that keratinized comes with this ability to be waterproof. There's a few more facts to add to that, but that we'll add it when we get to skin. But otherwise, that is generally what the difference is between the two. Non-keratinized found in the inner surface of things, inner lining of things, on the inside of the body, and keratinized is found on the outside. We don't really need this texture, skin texture in our throat, in our esophagus, in our rectum. We do not need that texture. We're going to keep it very simple today. And so how to know when you're looking at either or, right? Because you're going to have to make a comparison between these two. If they show you these two pictures, you got to know which one's keratinized and which one's non-keratinized. And the answer fits, it's really easy to remember. You can hold on to this story. That these cells are in a way like little kamikaze cells, right? Like they're going to sort of like make more copies of themselves, go to the surface, and then if your friction, whether again the vagina, the anus, or the esophagus or the throat, that whenever they encounter friction, they're going to go along with it and they die, right? They go away. They serve as sort of little suicide cells in a way. Like they're going to eventually, their role is to die. And so they go with your substances. Now, there's a difference on the surface that's in contact with the apical side. When you look at the apical side of most non-keratinized, you can still see these little dots. What are these little dots supposed to represent? Nuclei, nucleus, right? And if you look at the skin, these cells, they die way before they encounter any friction. As they're going, you can see some barely faint, these faint nuclei, these little white structures. It's hard to stain these type of cells, but otherwise, as you see, they travel up. By the time they reach halfway, they're dead. All you're seeing is their plasma membrane and the inside, and then they eventually make their way out. And they're already dead even before they made their way to the surface. And so that's the biggest difference, is that on most of these pictures, you can still lightly see some nuclei all the way leading on the apical side, but when you compare it to the actual keratinized, you'll see that there's no more nuclei. They're just dead little cells. And these cells, what keratin is a protein. These cells, if you can use your imagination, let's pretend I'm a little squamous cell and I started off here. I'm alive and I'm me, I'm Luis, right? I'm traveling through, I don't know what's going to happen to me. But then all of a sudden, I start swallowing cement, right? I start swallowing cement. Use your imaginations. The more I keep going up, then eventually I just die off and I'm just a little brick. That's what's happening with these cells, is that they're consuming a protein that makes them solid, 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 and then they just die, and then their role is to serve as skin. Something that you slosh off and lose. As I touch everything, I'm losing squamous cells. You're all losing it on your clothes, and that's why, yes, I'll leave it like that. And so, really easy peasy, right? And yes. And so we usually, just to intro this material for skin, because that is part of your first lecture exam, is that you will see keratinized stratified squamous always has two layers, or two zones. I'm going to call them zones. Zones. You have alive and dead. Alive and dead. So when you look at this segment, 2 is going to be dead. Dead. Right? Dead. This segment's going to be dead cells. This segment where you still see nuclei is going to be alive, living cells. Living cells. And let me see. There's a better picture of this. 1 second. Here. And so this is the image that's a little more popular to use. You'll notice that this is not the basement membrane. This is the basement membrane. So we're going to have to draw it in here. This is going to be living. Switch to red. Thicker. This is going to be living. And the segment above, switching to yellow. Dead. A living zone and a dead zone. You can call it living layer, or dead zone, or living zone. And that essentially concludes everything that can possibly be asked. And then these questions here will ask you that very thing. It already has the arrows for you. Number 2 would be that dead zone. Number 3 would be the living zone. But otherwise, that's how you can always tell when you're looking at keratinized, is that the cells, there's no more nuclei. It looks like hairs in a way on a microscope. But here you can see the cells are still, you can still see some cells in the body. All right, connective tissues is the topic we're going to address next class. And we are going to jump straight to the back of this packet because there's only a few left for muscles. There's only 3 muscles types. So we're going to go to page where it's just category 3 muscles. Page 43, thank you. 43. All right, so remember now, everything you've learned about tissues so far has been about epithelial tissues. As we address muscle tissues, you don't care about basement membranes. You don't care about basal. You don't care about apical. You don't care about secretion and absorption. You don't care, that's it. And then you're really now completely scrambling, restarting a new topic. Muscle, you have 3 muscles you care about. You have heart, or cardiac muscle cells. This picture is a little better. Heart or cardiac muscle cells. Again, in our heart, we see cardiac muscles. Our skeletal muscles, everything that you see when you look at these models, all of this, everywhere that's connected to all the body, not including the heart, but everything else, these are all muscles. They're called skeletal muscles because they move your skeleton. And then, do not forget your digestive tract. To go through that digestive tract, your stomach has to squeeze things in order to move things through your digestive tract, including your small intestine, including your large intestine. They all have to squeeze the food through in order to move it through. And so that's considered smooth muscle. And the good news is this is the easiest part out of all, second easiest out of all the tissues. Now, when you look at them, there are some similarities that you're going to encounter and some differences. Skeletal muscle and cardiac muscle. And let me check your notes very quick. When you look at your skeletal muscle, flip to this page here. When you look at this, they're a little bit different. than regular cells. Remember that we've seen only cells that look like either squamous, cuboidal, or huge little rectangular cells, columnar cells. These cells, nothing like it. If you look at this, what do you think these dots are? Nucleus. And so these nuclei, you'll notice that a columnar cell is probably, if you compare it to the size of this, it's probably this big. That's how big a columnar cell is. Maybe even smaller than that. Might be even like this big. A columnar cell. I'll draw it on there for the recording. That a columnar cell would be like this small in comparison. These would be like a simple layer of columnar cells. And each one had a little individual nuclei inside. But in this instance, muscle cells are completely different. They fuse together and form huge cells. Huge cells. So this entire cylinder you're seeing here is a muscle cell. But we don't call them muscle cells. We typically call them muscle fibers. It's the same exact word. Muscle cell and muscle fiber are the same exact word. And the reason is, you all have some you know what, I don't want to get too complicated, just keep it simple. So here we look at this segment here. When you look at these muscle fiber cells, they each have, if you look very closely, do you see those lines? Those lines are called striations. Striations. And that's really how you're going to be able to distinguish between this and the other cells. That they have stripes or striations. And they are multinucleated, meaning that they have many nuclei. And this is because of the way that they're formed, that the more you exercise, as an example, you destroy the muscle, and then these cells come in to sort of replenish, replace, repair, and then they end up fusing with it, and then their nucleus gets stuck inside with it. And so they're a little bit more complicated, but otherwise that's why they're multinucleated, and they're very, very long. Striations, nothing new to add there. That's all it is, be able to recognize. And where do you find it? Attached to the bones of the skeleton. Nice and easy, right? They're called skeletal muscle for a reason. They're attached to the skeleton. Next, we have cardiac muscles. Cardiac muscle is going to be very similar in that it causes movement of substances, but the difference is that they are not so perfectly cylindrical. You saw that on the muscles, skeletal muscles, they all ran very parallel to each other. And in this case, they will look multi-branched. You're seeing, like, as we follow my pen here, I'll use for the recording. Here, as we're traveling through here, you can see we branch into one fiber in this direction, one fiber in that direction. You don't really see that with skeletal muscles. So branching is a key indicator. Now, what were those lines called in skeletal muscle? Striations. And so you can see striations as well on all of these. But there is one big difference. What do we notice about some of these striations? What do we notice? As we keep going, and then what happens when I hit this? What's there? There's a thicker one, like a very thick one. Skeletal muscle doesn't have that. Only cardiac muscle has these discs. These discs are called intercalated discs, as written on your slides up here. Intercalated discs. They have striations too, but again, cardiac muscle has both striations and intercalated discs. Intercalated discs. And that's it. That's how you're going to be able to distinguish it. I like this picture or this slide because you can stain them differently, but nothing changes. If you look closely, striations, 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 and then this band right here, intercalated disc. Striation, 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 intercalated disc. And it's just a band, like a different band color, right? You can see it clear on this segment right here. That's the best segment right there. And then you can stain it another color. And so you can even see those intercalated discs always are there. They're always very clear to see when you get your eyes adjust to the pictures. A little hard to see that zoomed in, but yeah. And that's it. And smooth muscle, this is the only picture you have of smooth muscle. There's nothing I can do to help you to really understand this other than you're going to see two directions. This that you're seeing traced in black That black tracing is going to be the cells themselves, right? This is one individual muscle cell. They're a little shorter than most cells, than the other muscle cells, and they have a nuclei. You can see it here, right? And here's our nuclei. Now this is also the same exact cell as this. It's the exact same thing, but what's the difference? Is that these cells are found in layers. That these cells will go in this direction, and these cells go in this direction. Look at my arms right now. These are the two of the same structures right now. This is my arm, this is my arm. But this is one viewpoint, and that's what you're seeing here. You're seeing this viewpoint. This structure is the same exact thing as this, but instead you're seeing it looking right at you. It's going straight at you. But it is the same cells. They're just going in two different directions. But it's the same thing. It's this, but you're just slicing it differently, and you're looking straight at it. So this is like a transverse cut, and this is a longitudinal or sagittal cut. Very difficult to make out in your brains, I feel personally. And so the best thing to do is just remember this example I'm using with my hands. This cell is like my arm. It's just in this orientation. This one is in this orientation, or again, this orientation, but they're the same structure at the end of the day. And so this is the only picture we have of smooth muscle. So if you see this exact picture, but it may be rotated, you know, you just have to be able to recognize, oh, that's smooth muscle. And where do we find smooth muscle? Digestive tract, in digestion. And congrats, that's smooth muscle. That's all there is to smooth muscle. Here's another picture where you're seeing the exact same thing. Two of the same structures. One is laid out like this. This one is laid out looking right at all of you. It's the same exact structure. I won't be able to put that on the recording, but that is the best way to help you for this. Because this type of muscle tissue is found in two directions, and we'll learn why in digestion. And that's it. These are the types of questions you might encounter. And I'll let you all work on that at home. And then the good news is we're hitting the last category, which is nervous tissue. And nervous tissue, there's only really two cells you need to learn from here, which is really nice to conclude the class. The nervous tissue, where do we encounter nervous tissue? What system first? In the nervous system. And what are some structures, what organs that we learned that are involved in the nervous system? The brain and spinal cord. This is what it's made out of. This is what your entire brain, what all the connections and the wiring in your brain are. They're made out of these large ones first. That's what I want you to draw your eyes to first. These large cells are called neurons. Neurons. We also have cells that are little supportive cells. These cells are the biggest, demanding, the divas of the body. They're very, very sensitive, delicate, and very specific. Most cells can adapt to most things and pretty rigid, rugged, but neurons are very demanding cells, and so they have assistants. And the assistive cells are called neuroglia. Neuroglia. And in this photo, you're seeing both of them. All of these large ones are your neurons. All these little tiny dots are your neuroglia. All those little dots. Those little dots are the nuclei of the neuroglia. Neuroglia. But the large, spider-looking ones are going to be your neurons. And so some terms around it are just going to be play off of words that you already know. You have your neuron. That's the name of the cell. And when you examine the actual body of a cell, instead of calling it, the inside of it is still called a plasma membrane. A cytoplasm. Cytoplasm. But the entire body of it, we refer to it as cell body. Cell body. And then they also have a nucleus. And then they have this little structure called the nucleolus, but I want you to do this. I want you to cross it off. Do not worry about it for the lab exam. You'll have to worry about it in lecture, but I just think it's too out of place right now. So don't worry about nucleolus on your lab exam. And that's it. There's nothing else to add. There is nothing else to add. be able to answer these questions there. All right. And that summarizes 3. So the only one that we didn't cover is connective tissue. I do have a recording on connective tissues that I can share with all of you if you want a head start. And it'll also be our lecture topic for next week as well. So if you want to just get a head start on connective tissue, I'll make sure to put that link underneath the connective tissue section. And this is the first time I recorded in lab, so I'll find a way to organize it onto your Canvas. I'll have that done by today. And then I'll send an announcement as to how to find the recordings in order with your lecture notes. So it's really easy to just click with your notes, and it's just a little more streamlined. So I will see you all.